It's my pleasure today to introduce our second speaker of today's webinar. And our second speaker is Kristina Klobucar. And Kristina is a PhD candidate in the lab of Eric Brown at McMaster University. Kristina is a graduate of McMaster and has been working with Eric for a little while now. And she's going to be talking to us today about some of the novel antibacterial drug discovery work that's going on, in particular, focusing on drugs that are going to target gram negatives. So Kristina, without further ado, the floor is yours.
Thank you to the webinar organizers for inviting me to share my work here with you today. So I'll be talking to you about a chemical screening platform, which we at the Brown Lab have used to uncover new probes of the gram-negative outer membrane. So as many people are aware, the antibiotic resistance crisis is on the rise, and there is a need for new ways to treat resistant bacterial infections. So in 2017, the World Health Organization put out a list of priority pathogens which pose the greatest risk to human health and for which new treatments are urgently needed. So that list was divided into critical, high, and medium priority categories. And of the 12 groups of bacteria on this list, 9 were actually gram-negative. And these gram-negative bacteria, without even having any resistance genes, they themselves are intrinsically more resistant to antibiotics simply due to their cellular structure. So the structure that makes gram-negative bacteria different from gram-positives is their cell envelope, which is shown here. So you have a cytoplasmic or inner membrane consisting of a phospholipid bilayer, a thin layer of peptidoglycan within the periplasmic space, and an outer membrane, which is an asymmetric bilayer consisting of phospholipids on the inner leaflet and lipopolysaccharide, or LPS, on the outer leaflet. And this outer membrane acts as a permeability barrier to prevent toxins from the environment from entering into the cell. So due to this outer membrane, the chemical matter that's capable of cell penetration in gram-negatives is very limited. So for example, many antibiotics which are effective against gram-positive bacteria are ineffective against gram-negatives due to the inability to permeate across the outer membrane. And this is particularly true for antibiotics which are large in size, so greater than about 600 Daltons. And these antibiotics often require very high concentrations to produce any detectable growth inhibitory effect on gram-negative bacteria. So in particular, antibiotics such as erythromycin, vancomycin, novobiacin, rifampicin, and linezolid all have very high minimum inhibitory concentrations, or MICs, against gram-negative bacteria with an intact outer membrane. So it's important for us to find chemical probes that are capable of permeabilizing the outer membrane, as these kinds of molecules would have broad potential for outer membrane permeability studies and could potentially be used as adjuvants for treating gram-negative infections with existing antibiotics that are already clinically used. So in an effort to discover new compounds that are capable of perturbing the gram-negative outer membrane, the Brown Lab developed an unconventional screening platform to enrich for non-lethal molecules that increase the permeability of the outer membrane to large scaffold antibiotics. So I don't really have time to get into the complex biology that form the basis of this screen, but if you're interested, these papers here are a good read, and I'm happy to chat about it later if anyone is interested. But essentially, we know that if you grow E. coli at a lethal concentration of vancomycin at 15 degrees, so this is a temperature at which vancomycin is effective against E. coli, you can get suppressors of this activity by having deletions in outer membrane related genes. So notably, a lot of these deletion strains were in lipopolysaccharide biosynthesis. So in theory, if we screen for compounds which lead to growth in the presence of a lethal concentration of vancomycin at 15 degrees, we would expect to find compounds that target LPS or the outer membrane in some way. So Dr. John Stokes, who was a former PhD student in our lab, performed a pilot screen of about 1,400 previously approved drugs looking for vancomycin antagonism at 15 degrees. And he discovered that the compound pentamidine, shown here, was a hit. And here we have a checkerboard broth microdilution assay, which is used to test for interactions between two compounds. So on the x-axis, we have twofold dilutions of pentamidine, and on the y-axis, we have twofold dilutions of rifampicin, which is one of those large scaffold or gram-positive targeting antibiotics. And this was performed in a lab strain of E. coli. So the darker blue color represents more bacterial growth and white is no growth. And you can see that there is this staircase pattern in the checkerboard assay, which indicates synergy between pentamidine and rifampicin. Pentamidine was also found to potentiate the activity of novobiacin and erythromycin. And this was due to a physical disruption of the outer membrane rather than acting on an enzymatic target in outer membrane biosynthesis. So clearly screening for vancomycin antagonism in the cold gives us these types of potentiator molecules that we're interested in. But we wanted to cast the net wider than just this small pilot screen. So Dr. J.P. Cote, a former postdoc in the lab, screened a library of about 140,000 compounds, and these were diverse synthetic compounds, 100 times larger in size than the pilot screen. And he was looking for growth in the presence of 16 micrograms per mil of vancomycin at 15 degrees. So from that screen, he found 39 active compounds, and of those, 17 were resupplied based on availability and structural diversity. So from those 17, they were tested for synergy or ability to potentiate rifampicin at 37 degrees, and this resulted in 7 compounds. So of these, we chose to focus on 2, and unfortunately, I'm not able to share the structures of these compounds today as this work is still unpublished, but I can say that these compounds are not cationic peptides like most of the existing potentiator molecules that are just derivatives of polymyxins. So here we can see compound 1 is synergistic with rifampicin, novobiacin, erythromycin, and linezolid in E. coli. And a similar profile of potentiation is observed for compound 2. We also tested some clinical multi-drug resistant gram-negatives. So Klebsiella pneumonia in purple, Acinetobacter baumannii in red, and Pseudomonas aeruginosa in green. So compound 1 was able to potentiate rifampicin in Klebsiella and Acinetobacter, but not Pseudomonas, while compound 2 was effective against all 3 of these pathogens. So this was really exciting, as we can see that these 2 compounds potentiate antibiotics, which are typically ineffective against gram-negatives with an intact outer membrane. So we wanted to confirm whether this potentiation is due to outer membrane disruption as we might expect. And to do this, I used a lysozyme assay. So lysozyme is about a 14 kilodalton protein that hydrolyzes peptidoglycan. However, it cannot pass through the outer membrane of gram negatives. But if you add a compound to the cells, which increases the permeability of the outer membrane, this allows lysozyme to pass through and reach its target of peptidoglycan, resulting in cell lysis. So you can see here on the y-axis, we have cell lysis by lysozyme, and our control, which is just lysozyme and cells, shows very little cell lysis on its own. But if you expose cells to a subinhibitory concentration of a known outer membrane perturbant like SPR741, this results in an increase in cell lysis by lysozyme. And this was true as well for both compounds 1 and 2. Another method by which we investigated the effects of these compounds on the outer membrane was using atomic force microscopy, or AFM, and this was performed by Dr. Sean French in our lab. So basically, AFM uses a probe which scans the surface of the cell, and you can detect changes to the topology of the cell surface. So here we have untreated cells with whole cells in the top panel and the 3D topology of the surface on the bottom. And these cells here were grown in increasing concentrations of compound 2. So as you can see, by looking at the surface topology, adding more of the compound 2 resulted in increased undulations in the cell surface, and also resulted in these wide crevices being formed. And the same is shown for compound 1 below. So as you can see, increasing the concentration of compound 1 led to a more rough surface. However, for compound 1, we actually noticed that at 128 micrograms per mil, there were these vesicles or blebs forming on the cell surface. And if you zoom into a region of the cell that has these blebs, the surface is much more rough. However, a region that doesn't have as many of these blebs is more smooth. And it actually seems like this drug is perturbing the membrane in such a way that the cells need to get rid of these regions of the membrane and are shedding these vesicles. So since LPS is a major component of the outer leaflet of the gram-negative outer membrane, we hypothesized that these compounds are binding to LPS in order to perturb the integrity of the outer membrane. So the structure of LPS is shown here. We have a lipid A KDO2 region, as well as inner and outer core oligosaccharides. And typically there are also O antigen repeats. However, these are not present in E. coli K12, which is what we worked with. So to test for LPS binding, I used a Bodipi cadaverine probe, which is fluorescent. And Bodipi cadaverine is known to bind to LPS, specifically to that lipid A portion of the molecule. And this results in a quenching of its fluorescence. But if you add in a compound or drug which also binds to LPS, this results in displacement of that Bodipi cadaverine probe and leads to increased fluorescence intensity. So here you can see fluorescence intensity on the y-axis, and our positive control, SPR741, which is known to bind to LPS. So we have concentrations from 0 to 128 micrograms per mil from left to right. And the arrow just denotes the concentration of SPR741 required to potentiate 1 microgram per mil of rifampicin. So we can see, as we expect for a compound that binds LPS, you get a dose-dependent increase in fluorescence intensity. Compound 2 shows similar levels of fluorescence intensity as SPR741. As well, compound 1 does show a dose-dependent increase in fluorescence intensity. It is not as strong as the other two compounds, reaching a much lower amplitude. However, both compounds do seem to be binding LPS based on this, albeit compound 1 seems to be a much weaker binder. And one concern about compounds which act on the outer membrane is that oftentimes they're also active on phospholipid membranes in general. So we were interested in whether these compounds would be able to lyse red blood cells, which would be an indicator of cytotoxicity. And this experiment was performed by Jessica Wallace from the Bowdish lab. And as you can see at the concentrations used to potentiate, so that's 32 micrograms per mil of compound 1 and 2 micrograms per mil of compound 2, you get minimal to no hemolysis of sheep red blood cells. So that's a promising start for these compounds. So to summarize, we performed a high throughput screen looking for the growth of E. coli in the presence of vancomycin at 15 degrees and found 2 compounds which potentiate large scaffold antibiotics in gram-negative bacteria. This potentiation was due to these compounds physically disrupting the outer membrane through interactions with LPS. And these compounds are found to be non-hemolytic at potentiation concentrations. So we are currently still assessing whether these compounds would make suitable leads in the drug discovery process and whether they would be able to act as adjuvants for large scaffold antibiotics in vivo. However, if it doesn't work out, these compounds will still be newly characterized probes of the outer membrane and can further research on this outer membrane structure and biology, as well as potentially be used for studying intracellular processes, which are made difficult by this barrier. So with that, I would like to acknowledge my supervisor, Dr. Eric Brown, and all of the folks who are involved in this project. I was only able to show a snippet of the work done, but we had many people working on this from the Brown Lab, as well as the Magdalen, Bowdish, and Whitfield labs, and our collaborators from the Broad Institute. I'd also like to acknowledge our funding sources shown here, and thank you for listening, and I'd be happy to take any questions.